And then that is a surface that faces the shadowing source. It seems to me that the shadowing came from the bottom. Okay? And the reason that I believe that for sure is if I look at the small grainy particles that can only be proteins, and they have to stick out, and I always see the stain on the bottom side. So it seems like this is shadowed from the bottom. Does that make sense? It's interesting to look at fracturing images. So they give really high resolution information about the real structure of organelles and membranes. And it takes a little bit of practice to look at them and understand what's going on. So that's freeze fracture. So, this was the transmission EM. There's another form of EM called the scanning EM. The scanning EM is used much less in cell biology. Nevertheless, it is sometimes used and it can be interesting. So if this is the path in a transmission EM where we have a source, analogous to the light source in the light microscope, condenser lens, objective lens, projector lens, and somewhere we form the image. What happens in the scanning EM is a little bit different. It's more like a confocal laser microscope, the way the image is formed. So the top of the scanning EM is actually the same sort of as the transmission EM in that there's an electron source. That typically is a piece of tungsten that's heated up and electrons jump out, get out. They're captured in an electric field and they're accelerated. That's the same that happens actually in the transmission EM also. The acceleration of electrons is very important. Then they're focused, so you have a system of focusing lenses. And this focuses this primary beam of electrons on one part of our object, of our specimen. And the position of that focusing point is changed over time. That is the scanning process. It's very similar to the fluorescence laser scanning microscope. So how do we get an image? We get an image because these primary electrons, they hit the specimen, and the specimen is always stained again with heavy metals. And then what happens is the primary electrons, which have a lot of energy, bounce into the heavy metal electron sphere, and they kick out a couple of electrons from that specimen. And we call that the secondary electrons. They have a low speed, and we have a detector, which is a positively charged object, and it captures those secondary electrons. So electrons that are kicked out are captured by a detector. That gives a current. That current is detected by a computer, and it matches this as a brightness together with the position of the scan for the primary electrons. So we get an image formed by means of a computer, if you want. So it's the scanning principle. So electrons that hit the specimen here away from the detector have a low probability of reaching that detector. But electrons that hit the specimen close to the detector on this side have a good probability of hitting that detector. That gives a current that is read out as brightness. So the image appears as if it's brightly illuminated from a position that corresponds to the detector. That's how the image is formed in a scanning EM. Let's look at one or two examples of three. So here we have our friend, the bacteriophage T4, one of the T-even phages, as we call it. This is a virus that infects bacteria. So here we have a very high resolution image. This little viral particle was covered with heavy metals in order to make it visible. So where is the detector? What would you say? I think it is top left for you. What did you say? Because it seems as if we have snapped a picture here of an object that was illuminated from the top left. So that's the position of the detector. Electrons that hit on this side of our bacteriophage have a lower probability of reaching that detector. Not so bright, not so much current. Electrons that were kicked out by the primary beam here, where I'm right now with my cursor, have a high probability of reaching that detector. Okay, we can get very high resolution also with scanning EM, but we have to remember we're always looking at the metal that covers the biological material. So here we have fibers of collagen. Collagen is a protein that is in skin and I think also in hairs. It's a sort of a connective tissue type of material. There are many types of collagen, by the way. You can see a very high resolution of these protein structures here. Kind of rings in there. You see some dirt on there even. So where was the detector for the collagen fibers? At the bottom, at the top? At the top. Would you think more to the right, more to the left? I think it's top right. That's what I think. Here we have an insect head, a very scary looking insect head. Much lower magnification here. This is very high magnification. Whereas the detector seems to be a little bit on the left. All good? So understand how the images are formed, and you know what you're looking at, and you know the limitations. I'm going to follow up a little bit later in the class with some examples of different pictures, and I'm going to ask you some questions about how they might be formed. And one of the tasks that you will do will also include an exercise that really lets you look at different microscopy images. And you have to tell me something about how they were formed. Okay, that was that. We still have some time. We still have some time. Let's get a start here on my final. Why are you not looking? Okay, so we're looking at different methods, and we've looked at cell culturing, structural methods. Then we need to talk about biochemistry and fractionation of structures and molecules. We'll get to it later. Now I want to talk a little bit about the use of molecular biology. So, molecular biology, what is that? Molecular biologists are what we can call the gene jockeys. They play with DNA, and they play with RNA. I'm a little bit of a gene jockey, but not so much. So this part of biology we call molecular genetics, looking at genetics at the DNA level and the RNA level, and it's important often for the study of gene expression. So we'll talk about that a little bit. I want to summarize a couple of methods. An important reason is that next Monday in the lab we'll be using some of them. So, what do molecular biologists do? They use DNA that they have cloned to study what genes look like, how they're organized, and maybe how they are expressed. We are interested in the mechanism of transcription and translation, especially asking the question, where and when is a gene expressed? And ultimately, we hope to gain an idea about the function of proteins encoded by the genes that we are interested in. Now, in order to clone a gene and get the piece of DNA that you are interested in, that is actually typically the biggest challenge. The challenge is much smaller today than it was 15 years ago. And cloning a gene was a big feat. That was really very impressive if you could do that. Nowadays, because people sequence genomes rapidly, entire genomes, we often do not have to do so much cloning ourselves anymore. So how we identify a gene, we'll get to that a little bit later in the class. It is interesting to think about that. And what really is interesting, I think, is to let you think about how you can use the biological context of an organism in order to figure out how its genes function. Often there's a connection there. So right now, I just want to go over some applications. Say you have cloned your gene. So what can we do? Okay, one thing we can do is amplify that DNA sequence in order to figure out if it's there and in order to visualize it. And a commonly used method is the polymerase chain reaction, or PCR. So why do you use that? If you want to obtain a specific DNA fragment and you want to analyze it. So how do you do that? Let's imagine we have a target piece of DNA. This could be a piece of DNA in yeast. Now if we know the sequence of that DNA, we can have synthesized by our friends a small stretch of DNA that we call a primer. And we have two of those. And these primers can bind to the target DNA. But in order for the primer to bind, we first have to make the DNA single-stranded. So our target DNA has to be made single-stranded. How can you do that? DNA is kept together by means of hydrogen bonds. And if we heat up the sample, the hydrogen bonds become relatively weak, thermal motion takes over, and the strands separate. We call that melting. So the two strands of DNA can be separated, can be melted. It takes a temperature of something like 85 to 90 degrees Celsius to do that. And in a PCR reaction, we often use 94 to 96 degrees just to be sure. So we separate by heating up. Then we cool the sample down. And the primers, which we have in excess, in molar excess, can then find their target sequence. Microphone is up. I need to shout for 2 minutes. The primers find their target sequence and they bind there. We call that hybridization. Then the primer that is bound to the target sequence can be extended by the DNA polymerase in the presence of the 4 subunit nucleotides. So then we have formed a copy. We had 1, and now we have 2 copies of that DNA. And we can repeat this cycle. So we go from 1 copy to 2, to 4, to 8, on and on. In the end, you can have millions. And a single DNA molecule cannot be visualized, but millions of DNA molecules of the same size can easily be visualized. So later, we will analyze these products by electrophoresis. Now, in order for this to work, we need a DNA polymerase that can withstand that melting step, that high temperature. And therefore, we use a DNA polymerase that is isolated from bacteria that live in the hot springs in Yellowstone. And we call that the TAC polymerase, TAC for Thermophilus aquaticus. You know, that's enough. I will see you on Monday.